Hi, everyone. So first, I would like to thank Diane, Sarah, and Nicole so much for the kind invitation and introduction. And I'm very honored to kick off MPG Primer again after the summer break with our recent paper. So I'd like to use this opportunity also to thank my co-authors who I've been very fortunate to work with in this project. So while I unfortunately cannot be at the Broad today, some of them might be. So if you wanted to discuss further, maybe you can actually grab them there. The data of this talk comes from the FIMM research project. So disclosure slide here, this is funded by a lot of industry partners, but they had no involvement in the design of that study. So this being a primer, let me briefly introduce the FIMM research project to you that aims to utilize genetic strategies to understand disease mechanisms, linking genotype data with digital healthcare data in the form of electronic health records. And the study is now really in the final recruiting stage. So the second FIMM 2, basically the second phase of that study, has been finalized, over fulfilling the goal, genotyping 10% of the Finnish population. The data freezes of this talk will, however, be from previous releases, somewhat smaller. That the recruitment phase is finalized does not mean, of course, that FIMM will stop, but it actually currently enters the next phase, focusing on disease mechanisms and disease progression for the next 4 years, which has just launched. So stay tuned for updates from this very exciting project. So why is it actually why is Finland actually a really great country to do this type of research? Of course, that's predominantly coming from the excellent infrastructure with biobanks, universal healthcare systems, personal identity codes of the electronic healthcare data and so forth. But there are also some unique features of the Finnish population that make it quite suitable. And I'm going to give you a bit more insights now. So at large population sizes, usually natural selection lowers the frequency of deleterious genetic variants, but sometimes populations can undergo drastic reductions in sizes, and that's usually called a population bottleneck. And what that means is that the number of rare variants is consequently reduced, as is the number of individuals. And what that also means is that some of those rare variants, being previously ultra rare, will just by chance rise in frequency. Stochastically, some of them will also be deleterious, so having some effects on disease. So in Finland, that's exactly what happened. So there have been multiple bottlenecks, and they have not been that long ago. So the time has not been sufficient to remove some of those deleterious variants again, or lower their frequency. Of course, what also has to happen is some specific geographic and thus genetic isolation. And in consequence, there are multiple diseases that only exist in Finland, and those are predominantly from the recessive inheritance space. And this has been previously described and explored very carefully, so that these diseases are commonly referred to as the Finnish disease heritage. What does that mean specifically in terms of variants? So previous studies have identified that a subset of deleterious variants are enriched in Finland, specifically in that intermediate to rare frequency range. And that, of course, gives us more power to study their effects on disease. And as is also common for populations with recent bottlenecks, they are characterized by higher rates of homozygosity, and this is also the case in Finland. And this, of course, increases the chance occurrence of two potentially deleterious variants in homozygous state, and thus it's not surprising that those founder diseases are mainly having a recessive inheritance. So we were wondering here if that does not only apply to deleterious variants in general, but maybe also specifically to disease-causing variants. So we set out to investigate whether there was maybe a subset of disease-causing variants more frequent in Finnish than non-Finnish Europeans, specifically in those genes annotated with recessive inheritance in OMIM. So we investigated those variants in NOMAD in 10,000 Finnish in comparison to 10,000 non-Finnish Europeans. And we did indeed find that, as expected, we have fewer variants in Finns, but they are at higher frequencies than the non-Finnish Europeans. So that led us to hope that we might also be powered to potentially identify novel variants with homozygous effects on disease. Consequently, we set out to do a phenome-wide association study, a GWAS, of homozygous effects in FinnGen, analyzing 44,000 or more than 44,000 coding variants, and they're specifically investigating their effects on more than 2,000 different disease phenotypes. And we might not detect those purely homozygous effects with the standard additive GWAS model that assumes additive effects, as you can see here. So that's why we set out to search for homozygous effects. So as you can see here, the homozygous effect on disease is there's only a homozygous effect on disease, but not in the heterozygotes. And we implemented that with a very well-known method, SAGE, by Wei Zhu et al. And in the next slides, I'm going to compare the results of those two different models. So here you can see on the x-axis the additive model, and on the y-axis the p-values of the recessive model of all significant variant phenotype associations. So if any association was genome-wide significant in either the additive or the recessive model. And of course, what the main result is here, what has been shown previously, but of course, so therefore it's also what we expect, is that for most variant phenotype association, the additive model fits best. However, for some variant phenotype associations, and we've colored here in blue those instances where the recessive model fits substantially better than the additive model. Specifically, the recessive model's p-values is 2 orders of magnitude lower than the additive model's p-values. And one example here is the GJB2 gene associated with hearing loss, which is significant only with the recessive model. So we would not have picked it up with the standard additive model at all. So next, the next question we wanted to investigate, are any of the recessive associations in known recessive disease genes? So to answer that, we broke them down by known Mendelian inheritance according to OMIM. And you can see quite clearly that we found no recessive associations in genes annotated with dominant inheritance, but they were really enriched in recessive or dominant as well as recessive genes. So that's, of course, as we would expect. And that becomes maybe even more clear when we break the associations down to independent loci. And some of you may recognize some of these known disease-causing genes. So I'm highlighting here again GJB2 that I mentioned earlier. I'm also highlighting NPHS1 for the reason that it's actually a Finnish disease heritage gene associated with a Finnish congenital nephrosis. So that's, again, a disease that is really only really observed in Finland. And reassuringly, it's not only the genes, but also the actual variants that have previously been described disease causing. So some 9 out of the 13 homozygous associations were previously reported disease causing. So that's very much reassuring that we are finding what we expect to find. So I hope that I can then convince you that we also have some power to find novel recessive associations. And I'd like to share with you three examples here. If there are no further questions up to now. So the first example that I would like to share with you is a variant in the CASP7 gene associated with cataract. And cataract is a condition that's where the eye lens becomes clouded, causing vision loss. And it's really the leading cause of vision loss worldwide, specifically affecting low middle income countries, because surgery is really the only effective treatment option so far, and it's not really accessible everywhere. And we now find a missense variant associated with cataract that is really showing a recessive inheritance pattern. So you can quite clearly see that the disease onset is much earlier in homozygous carriers. By retirement age of 63 in Finland, about half of homozygous carriers had developed cataracts. And to also what we also found quite nice is that this is really a specific association. So there are no other phenotype associations in homozygous carriers. And at least I'm not aware, to the best of my knowledge, that that there are other Mendelian sized effects, genetic effects on adult onset cataract before we found that variant. The next example that I'd like to share with you is a gene called C1090 associated with hearing loss, and here the effect is even larger, so even more sort of Mendelian sized effect with an even earlier disease onset. I should also say that of course we have the limitation of electronic health records, so it's the time of diagnosis that we can record, but the disease onset can of course happen already much earlier. And again, there are no other frequent diagnoses of homozygous carriers other than sensory neural hearing loss. So it's a really specific association for that phenotype. So next, I'd like to share with you a novel locus with a recessive effect on female infertility. The coding variant we found in the PKH1DL1 gene is having an effect on splicing. So it's a splice variant leading to loss of function of that gene. However, I have to say that it's a little bit unclear still what the causal variant at that locus is, because back then, it seemed to us that EBEC9 would be a more plausible candidate, given the fine mapping results and effect sizes. But now with the newest data releases, it doesn't look that way anymore. So I would sort of just disclose that we're not super sure at that point in time. But the locus is very clear, and of course, I can show you also the validations here that we replicated in the next FINGen release R6, where you can see a very nice confirmation of our associations in both variants here. And digging a little bit deeper into that phenotype, we would expect, of course, for a phenotype like female infertility, that affected women and carriers might have fewer offspring, fewer children. And this is exactly what we observe. So the number of children in homozygous carriers are on average 1.7 compared to 2.0 in wild type carriers. And the affected women who are also diagnosed with infertility, compared to all women diagnosed with infertility, bi-allelic homozygous carriers actually are even more severely affected in that they have their first child again significantly later. So we also aim to replicate recessive associations in other data sets. And so we used the later FINGEN release 6 for that, and also the UK Biobank, of course. However, it was actually a little bit tricky to validate all of our data, all of our associations due to the Finnish population's isolation, which means that most of the variants we found were actually enriched in Finland and therefore at low frequencies in other populations. However, we could still validate some of them in the UK Biobank and also in FINGEN Data 3.6. So in total, we could then validate 20 out of the 24 homozygous associations. And these are the novel associations that I just described to you. And these ones we could also, at least 2 of them were at sufficient minor allele frequency so that we could also replicate them in the UK Biobank. So next we were wondering how many recessive association would actually be missed if we only used the additive model and not the recessive model. So we performed simulations, 100,000 simulations of recessive associations resembling our data. So specifically, that means a population of 200,000 variant in Hardy-Weinberg equilibrium. And then we investigated effects at different allele frequencies. And as you can see here, that the recessive model at low minor allele frequencies can uncover associations that are completely missed or almost completely missed by the additive model in that frequency range. But as we move up in minor allele frequencies, we can really also with the additive model find all those recessive associations quite nicely. So this is really reflected also in our data, where we could indeed only find 13 out of the 20 recessive associations with the additive model, meaning that we did we would not have found them if we just ran if we had just ran the additive model. So that leads us to speculate a little bit if it would make sense to run a recessive model more frequently, in addition to an additive GWAS model that's commonly used and very rarely done. Of course, multiple studies have found little contribution of non-additive effects in common variants. So this has been investigated many times previously, and it still holds true today. On the other hand, there are plenty of examples for Mendelian inheritance of recessive and dominant in coding variants, and with better imputation, the genotype data will often more and more include rare coding variants, just as our data set does. data sets become also larger and larger. So it is worth considering recessive models in GWAS, and we've also seen it happen more frequently. So next, changing gears a little bit, if there are no question on that last part. We next aim to investigate mono and bi-allelic effects of known disease-causing variants in our data. So public variant databases such as ClinVar are really central for genetic diagnosis, but of course it's a community effort, so they're bound to be some errors every now and then. So we wanted to investigate if we, with our phenotype association studies on more than 2,000 disease phenotypes, could maybe enhance interpretation of variants in ClinVar. And we could actually pinpoint some very interesting examples where we could indeed say, well, we have in our data set for COL5A2, for example, specifically relevant joint disorders, or for PKD1 specifically relevant cystic kidney diseases. We have really thousands of disease individuals with such diseases, and we should really expect to find at least a nominal significant associations, and if we don't, then that's really a clear sign that such variants are not disease associated. While for other variants, we actually can confirm disease associations.
If you have questions, we welcome you to raise those, and you can use the microphone in the center of the room, or if you're not in the room, please post them online, as Nicole has mentioned.
So another aspect that we can also use our data to investigate is that we can dive deeper into mono and bi-allelic effects of disease variants, and contrast those effects that we find with the inheritance models that are annotated for such variants. And here we started exploring effects of disease-causing variants in genes with dominant inheritance. And what we found here mainly were variants in tumor suppressor genes. So let me show you a few examples here, beginning with a missense variant in OCA2, and that is clearly associated with an increased risk for skin cancer, as well as a missense variant in the MCR1 gene. And even though the genes are clearly annotated with dominant inheritance, I think it's very much clear that the pattern here is very much a dosage effect, resembling much more additive or let's say semi-dominant pattern of inheritance. Same or similar for a loss of function variant in the CHEK2 gene in association with cancer in general. Yeah, so that really leads us to question a little bit whether it makes sense to call those associations or those patterns dominant inheritance, or if it's actually a little bit more on the additive side here. And next, we wanted to explore also then the effects of disease-causing variants in genes with recessive inheritance. And we started off by investigating global disease effects, comparing to randomly sampled variants at same minor allele frequencies. And we were surprised to find effects in genes with recessive inheritance, likely pathogenic, as well as some of them associated to be of conflicting evidence. And that's, raises the question if heterozygous effects contributed potentially to that signal. It's also quite nicely aligned with some work that other people from the Broad, Paul Rue's lab did last year in the UK Biobank. And diving more deeper into what the variants are driving that, I'd like to share with you a disease-causing variant in the XPA gene. So it has been known that that this gene is associated, loss of function of that gene is associated with xeroderma pigmentosum. So that's a condition linked to childhood onset skin cancer. And what we find here is that in heterozygous state, there is an association with adult onset skin cancer, which makes, of course, which fits quite nicely that sort of additive pattern. It's really a related disease, and reassuringly, there is a different variant, different protein truncating variant in that same gene that also leads to actually a different type of skin cancer in a Japanese study with a similar odds ratio, which is a nice confirmation. Other variants that would have subtle heterozygous effects that we could recapitulate were one variant in the SERPINA1 gene that's known to also in heterozygous state have a very small effect on the risk of emphysema. There's also been some previous debate on heterozygous effects of variants in the NPHS1 gene causing nephrotic syndrome, which I've earlier mentioned. So we also observed an effect here. CASP7 is a novel disease gene that we found associated with cataract. Even there, we found a very modest, nominally significant heterozygous effects that we could also replicate after excluding compound heterozygous effects. And then also for GJB2. So it really seems like if you increase the data set more and more, there are more and more of these tiny heterozygous effects appearing in genes with recessive inheritance. So exploring unusual inheritance patterns more broadly, I'd like to share also some other what I think is a really cool variant, a missense variant in STN5A associated to severe arrhythmia, specifically like 6 sinus syndrome. And here we found that one mutation in our data was actually protective of cardiac arrhythmia. And we could actually validate that in the UK Biobank. Of course, it's again a Finnish enriched variant, so the UK Biobank validation is at a much smaller p-value, and of course the main phenotype here in cardiac arrhythmia is atrial fibrillation, but we're also finding those effects in some other cardiac arrhythmia phenotype. So we're currently writing a manuscript up, so stay tuned for some updates. Also, we see some patterns in ECG data fitting to those previously described loss of function effect in experimental data. A little off topic, we were also somewhat surprised to find quite many genome-wide associations for benign variants. And initially, we were somewhat surprised by that because benign variants are, of course, defined as not being the cause for a patient's disorder. So we thought maybe they were innocent bystanders, but fine mapping shows that 17 of them are even the top causal variant after fine mapping. So actually driving those signals. But then we thought again about it and realized that, of course, benign variants are defined as having no Mendelian effect, but that means they still, of course, can have weaker effects on related phenotypes, and that's something to maybe remind yourself again before using them regarding them as a sort of group of variants with neutral effects. So they're probably not so frequently neutral as we would intuitively expect. And an example of that is here the benign variant in DBH, protecting from hypertension. Quite fittingly, the DBH gene is associated with a disease with recessive inheritance, leading to severe hypotension caused by low levels of noradrenaline. And I think it's biologically then very plausible that the variant we found actually protects from hypertension in our data. So to conclude, we have large biobank data that we can use to find phenotype associations of Mendelian disease variants beyond the traditional labels and inheritance patterns of recessive or dominant, or maybe benign, looking a little bit beyond these labels. And the question is what the implications potentially could be if we have Mendelian variants with heterozygous and homozygous effects on the same or related diseases, because the traditional labels of dominant and recessive don't really adequately describe such patterns. Semi-dominant inheritance is mentioned in many of the guidelines, but it's not used in practice at all, neither in ClinVar nor in HPO. However, it may be worth thinking about if it can actually be beneficial to integrate the heterozygous or homozygous effects for variant interpretation. Lastly, we felt the need to harmonize the nomenclature, and here I'd like to thank Mark and Heidi, who have had lots of discussions about. So starting with the traditional recessive and dominant inheritance patterns in rare disease, we and of course others found that there are many things that are falling a little bit in that in-between space, and linking that to the more complex disease genetics or complex genetics space. We thought it's
Henrike, how common are the protective variants versus the disease-causing variants that you found? Like, were there many, or
good question. We found that there were predominantly more disease-causing, so variants that increase disease risk versus variants that protect from disease risk, even for the benign for the benign variants. So that's one of the things that we also suspected, that maybe benign variants are disease associated, but that means they much more frequently protect, and they do protect sometimes, but more of them actually also slightly increase disease risk.
Okay, thank you.
So in summary, I hope I could convince you that our data from FinnGen gives us a great opportunity to study recessive Mendelian associations at population scale, enabling us to find some already known, but also some novel disease associations across a large spectrum of phenotypes. And we can also use our data to investigate disease associations of variants with Mendelian effects that do not fit really those traditional clinical genetic labels of recessive and dominant or maybe benign. I'd like to conclude by thanking the many people in the FinGen consortium, specifically to Arno and Mark for leadership and mentorship, and also the team leaders, specifically also Mitya and Juha, all FinGen participants. And then I'd like to thank you so much for your attention.
Thank you so much, Henrike, for a wonderful, thought-provoking talk. And thanks everyone for joining us, whether in person or online, and we will see you again soon. Thank you.
Thank you so much. Take care.